muscle and plain muscles, unlike the skeletal muscle. Okay, in detail we'll be discussing about this smooth muscle in this lecture. If you see the structure of the smooth muscle, and as like your skeletal muscle, we have contractile proteins are there, actin, myosin, troponin and tropomyosin. Actin and tropomyosin forming the thin filaments, the otherwise myosin forms the thick filament. This is a similarity as like that of your skeletal muscle. In place of the Z lines are absent. Z lines are replaced by the dense bodies. Here you can see the Z lines, you can see over here. Z lines are replaced by the dense bodies, and I will just take here the color for writing the picture. Yeah, I have selected the pen. Now I'll pull my diagram down. So this is not yet selected yet. Okay, it is selected right now. Coming to here, these are the dense bodies. Dense bodies are the structures. Here I'm rotating it, and the dense bodies to which the myosin and actin filaments are attached. The dense bodies in turn they attaches to the sarcolemma. So sarcolemma is a membrane of the smooth muscle fiber, and finally the dense bodies are finally attaches to the sarcolemma. So this is the structure, thin filaments, thick filaments, so many gap junctions are there, like cardiac muscle, nucleus, elongated nucleus, dense bodies and intermediate filaments are there. The difference between these two pictures are, this is a contracted state, and it is called relaxed state of the picture. Okay, then we'll go for the next one, next slide. In the next slide, now we'll talk about the types of the smooth muscle. The smooth muscle is of two types. Okay, one is called single unit. One is called single unit smooth muscle fiber. Two type of smooth muscle fibers are there. Single unit smooth muscle fiber. The other one known as multi-unit smooth muscle fiber. The single unit smooth muscle fiber is also called visceral muscle fiber. Under few headings, I would like to finish the differences. Number one, it has got your low resistance bridges are there. Low resistance bridges. The low resistance bridges are the one and which are required for the functional syncytium. The smooth muscle fiber function like a unit or contract like a unit, but whereas your multi-unit smooth muscle fiber is non-syncytial in nature, and the contractions are called discrete, localized and fine. Okay, they are called localized and discrete and fine contractions. You can see this is the first difference. Now we'll see the second difference is sites where these single unit smooth muscle fibers are located. They are located in the wall of the hollow viscera, like examples GIT, uterus and ureter, bronchi and urinary bladder, etc. These are the areas areas the single unit smooth muscle fibers are located. And here these are located in the iris and the ciliary muscle, not only iris and ciliary muscle, muscle of the eye, piloerector muscle of the skin, muscles of the blood vessels. These are the areas where multi-unit smooth muscle fibers are located. Now coming to third one. The third difference is characterized by pacemaker. Okay, these are characterized by pacemaker. We know the pacemaker are the cells, pacemakers are the one and which generate their own impulse. We could see them in the GIT, gut cells, and the innervation modified by extrinsic innervation. It is innervated by extrinsic nervous system, and here the innervation is, it is richly innervated, and each muscle fiber has got its own nerve supply. Each fiber has its own innervation. But here group of the muscle cell, group of the fibers will have one nerve supply, and the contractions are rhythmic. The rhythmic contraction and relaxations are there. Rhythmic contraction and relaxation, relaxation is seen over here. And the multi-unit muscle fibers, when the stimulus is applied and firing the repeated firing the action potential, which develop the irregular tetanic contractions, irregular tetanic contractions or tetanic contractions are developed. Tetanic contractions. So the final one, when the smooth muscle fiber is stretched, when it is stretched, it causes a tension, and active tension is developed in them. Active tension is developed in them. Here, these muscle fibers, they do not respond to stretch only. When here stretching, if it is stretched, active tension is developed. They do not respond to stretch. These are the striking differences between single unit and multi-unit smooth muscle fibers. Very important for MBBS examination point of view. Okay, so with this I have completed the types of the smooth muscle fibers. I can show you the differences over here. I just put my diagram somewhere around this side. Single unit smooth muscle fiber, a group of the fibers are innervated by a single neuron. But here, multi-unit smooth muscle fiber, every fiber is innervated by single separate nerve. That is the difference between smooth muscle and multi-unit smooth muscle fibers. So the same thing I have told you here. The same thing here I could show you. This is in the gut. In the gut you can see. This is the gut where group of the fibers innervating the single nerve, autonomic nerve. Okay, varicosity over here. But a multi-unit smooth muscle fiber, you can see in the eye ciliary muscle, and each single muscle fiber innervated by a single nerve system. Single neuron is innervating each nerve muscle fiber. Well, coming to the properties, under the properties of the smooth muscle, the property is very, very important. You can see electrical, mainly two properties can be seen over here: electrical property and mechanical property. Electrical property, single muscle twitch. When a stimulus is applied and after the applying of the stimulus, depolarization, okay, repolarization, then coming to isoelectric line and completion of both the activities is called single muscle twitch. And also mechanical property includes the contraction, which will have the latch bridge mechanism and plasticity. These two properties are very, very, very important, okay, under the properties. We'll see in detail what these properties are. The first property is, what is the first property? Property, electrical property. In a single muscle, single twitch, a stimulus is applied, a curve is formed. Electrical, under the electrical property, what happens is the average duration of this spike is varying from 25 to 50 millisecond. Later, the amplitude is very less and hardly it will reaches the isoelectric line. Okay, it is due to what we call it as action. It is due to nervous stimuli and leading to contraction. So, action potential that is developed in the smooth muscle fiber, it varies. We call it as minus 50 to minus 70 millivolt. It is unstable. Okay, it is due to intervening of pacemaker cells which have varying permeability to calcium ions and sodium ions. That is due to the intervention of this calcium entry causing to develop the varying action potential. There are three types of action potentials are formed. One is called spike potential. You can see the spike over there. Spike potential over there. Number two, spike potential initiated by slow wave rhythm. Number three, action potential with the plateau. All I will tell you now. So the first thing is spike potential. What is the spike potential? Here the action potential, we call it as action potential. And the spike potential is the one where depolarization of a smooth muscle fiber is dependent on influx of the calcium, not the sodium. The depolarization is brought about by nervous stimuli or hormones or neurotransmitters. So, and in this L type of the calcium channels on the membrane leading to influx of the calcium ions. Calcium entry causing the what is called your spike potential. So, spike potential initiated by slow wave rhythm. This is in a rhythmic fashion, slow waves are there. Rhythmic fashion, slow wave are there. And this is seen in the tissue that is containing pacemaker cells in the smooth muscle gut. Already I explained you. Spike potential appear this spike potential appear at the peak of the rhythmic contraction. Okay, these potentials are self-excitatory due to what is called opening of the calcium channels, which causes the depolarization already mentioned. Now this is I'm talking about this one even. Okay, these are pacemaker cells. Pacemaker cells are they are confining to the GIT. They produce the spike potential. The spontaneous electrical activity is seen in the visceral smooth muscle. It is completely different than comparing with the skeletal muscle. And I have shown you the spike already. It is a complete spike is formed. So minus 50 to isoelectric line and it overshoots and a spike is formed. A plateau is, there is a delay in repolarization. This delay is called plateau. This is due to prolonged exposure of large number of the calcium channels leading to sustained sustained presence of calcium intracellularly and causes a plateau at the time of the depolarization. So these are called slow wave. I told you slow wave rhythm, and this is what you call it as spike potential. Okay, slow wave rhythm, slow wave. It is one slow wave, and these are the spike. These are the spikes over slow wave. So the resting membrane potential could be the spike, could be the not stable, unstable, not unlike skeletal muscle, not minus 90 or minus 70 it is fixed. It varies minus 50 to minus 70 millivolt. What is the ionic basis for this? What is called ionic basis of the action potential in the smooth muscle fiber? The ionic basis is mainly due to opening of the calcium channel, voltage gated calcium channel, then the spike of the action potential. This is highest level of calcium around the isoelectric phase, and repolarization is due to opening of the calcium dependent potassium channels that leads to potassium efflux over there. So slow hyperpolarization, continued efflux of the potassium causes slow hyperpolarization, then the slow depolarization again, and closure of the calcium channel and opening of the calcium closure of the calcium channel and calcium dependent potassium channel causing the slow depolarization. These are the phases of the action potential, ionic basis of action potential. So now I can say here the same thing, voltage gated calcium channels, when they open up and action potential is developed, that is due to calcium influx and calcium concentration raises, opening of calcium dependent potassium channel, slow hyperpolarization, this line indicating, and then voltage gated calcium channel closes, then internal calcium concentration declines. So finally calcium dependent potassium channels also closes. Again it will come back to stage 1, voltage gated calcium channel open up. Again after forming one curve, and other curve will form in this fashion. So repeated depolarization and repolarization are developed by the events. This is what you call it as ionic basis of the action potential. So coming to the mechanical properties, and one of the property of the smooth muscle is the contraction. We have studied in the skeletal muscle also, when a muscle is in a what do you call a relaxed state, some sort of tone, some sort of what is called contractions remain in the muscle, and these are called tone or tonus. And see over there, we could see in this picture the relation between action potential and tension in the viscous smooth muscle. As the action potential forming, the tension also developed in the muscle fibers. Okay, this relation is nothing but producing the contraction, that is tonus or tone, one of the mechanical property of the smooth muscle. The other one, excitation contraction coupling, is very slow process comparing with the skeletal muscle fiber, and because the muscle starts contracting, why it is muscle contracting about 200 milliseconds after the start of the spike. Okay, so it is very delayed process and comparing with the skeletal muscle. So when you talk about excitation contraction coupling, here it is slow, because already I told you how delay it is. Unique feature here is a property of the plasticity. The meaning of the plasticity is, here there are two diagrams are there. One diagram representing the what is called length tension relationship in smooth muscle fiber, that is the property of the plasticity. See, as the volume increases from 0 to 200, 400, 600, 800, and the pressure also builds up, pressure also increases. So, and see over there, this is called filling curve. As we are, suppose imagine urinary bladder, as it is filling, and there will be a filling curve and emptying curve. And in case of the GIT, like stomach, you take, as you feed the food, and food with the volume of the food, the stomach size increases and will attain a new length after feeding, after filling it with a additional amount of the food. So this property is called the adaptation. Adapting to a new varying length from the existing length is known as the plasticity. It is all depends upon, the changes occurring in the actin filament, myosin filament, and the dense bodies. So this can be applicable to all skeletal muscle fibers, or including GIT and urinary bladder, maybe it is a ureter, whatever it is, okay, whatever the structure it is, it applies to all the structures. And this part is very important. This is asked many times in the examination. Events of contraction in the smooth muscle fiber. Contraction, how does the muscle is getting contracted? And suppose when you talk about a smooth muscle fiber, I just draw over here with elongated nucleus. It has got its own receptor. When acetylcholine binding with the receptor has, there are two types of the receptors, or their muscarinic receptor. The another one is known as muscarinic. This is a muscarinic receptor. There are two types of nicotinic and muscarinic receptors are there. It is which are present over here are muscarinic. When the acetylcholine binding with the and muscarinic receptor, intracellular intracellular calcium influx or increases calcium influx. The calcium influx into the cell, automatically calcium influx will be there into the cell. So the calcium and is binding with a protein called colmadilin, which activate the calmadilin dependent light chain kinase enzyme, one of the enzyme which is dependent on calmadilin. So immediately phosphorylation occurs, phosphorylation of the myosin head, that causes myosin activation of myosin head, binding of myosin to the actin, and the contraction process begins. The same thing, if the question asked in the examination, please write the steps what are involved here, what are there here, and explain. Self-explanation is needed. For a 5-mark answer, you can't just put the flow chart and expect 5 marks. Go better, self-explanation is very, very important. The relaxation is a completely opposite process, opposite or reverse process. So what is this? Calcium within the smooth muscle cell falls, calcium inside the smooth muscle cell falling or decreases, dephosphorylation of myosin by myosin light chain phosphate. Then dissociation of calcium from calmadulin and muscle relaxation occurs. Here one point to be noted is very, very important, is called latch bridge mechanism. Okay, this is a what we call it as a viva question in the examination. Okay, examiner asks you what is latch bridge mechanism. Generally during relaxation time, muscles sustain in a contraction state because myosin cross bridges, they remain attached to actin even after decrease in the intracellular calcium. Maybe and it lasts for minutes to hours to days also. This process, though calcium availability is not there, it still attaches with the actin, attaches with the myosin. This phenomenon is called latch bridge mechanism, one of the property, and very important mechanism, or it can be asked as a short notes also, very short notes. So you should aware of what exactly the latch bridge mechanism is. So now this picture shows you the nerve supply. In the first class, in the beginning of the class, I told you the innervation is a single unit, a group of the fibers are innervated by a single neuron. Multi-unit, all the individual nerve fiber supplied by individual neuron. I told you. So you see the nerve supply to the muscle cells here. And when you carefully observe inside the neurons, they have the vesicle with a neurotransmitter. Okay, so the neuron or the nerve with the many vesicles is called varicosity, which is autonomic in function. This is a single unit smooth muscle fiber, whereas multi-unit smooth muscle fiber, you have, it is like almost your what is that, neuromuscular junction, and same synaptic vesicle is there, neurotransmitter is released, smooth muscle is there, it is going and acting and binding with the receptor over the smooth muscle on the postganglionic axons of autonomic nervous system. Okay, this is how the propagation of action potential through the receptor. Gap junctions also occur. You know, gap junctions, the action potential goes very fast, in a rapid fashion, in a rapid manner it will. So once the action potential goes in a rapid fashion, it works, contracts like a unit, functional sensation. Thus that property it shows the smooth muscle fiber. Next, after this, we'll go to the another part called factors that affecting the membrane potential of the smooth muscle. Intestinal smooth muscle fiber. What factors affecting the membrane potential? There are three factors affecting the membrane potential. Number one, effect of catecholamines or catecholamine or stimulation of the sympathetic innervation. The sympathetic innervation, this is what I'm trying to say. Effect of catecholamine or stimulation of sympathetic innervation. And when the catecholamines, they act via both alpha and beta adrenergic receptors, which increases in calcium efflux that produce hyperpolarization of the membrane, decrease in spike frequency. These both activities decreases muscle tension and muscle relaxes. For relaxing the muscle tension, for relaxing the muscle, and the catecholamines or sympathetic stimulation is essential, which is depending on the calcium acting via alpha and beta adrenergic receptor. There is another one, acetylcholine or stimulation of cholinergic nerve. In reverse, in opposite with the above action, intracellular calcium influx causes membrane not hyperpolarization, depolarization, increase in spike frequency. Both will increases muscle tension and muscle contraction. So similar effects are seen with the cold and stretch stimuli also. These are the two effects what I have told you in the factors affecting the membrane potential of intestinal smooth muscle. The third one, effect of ions. Here, increase in calcium in the ECF increases sodium influx, increases height of the contraction. And barium, increase in the frequency and force of contraction is a direct effect, and increase in sodium in ECF, there is no effect at all. So intestinal smooth muscle potentials, intestine, I didn't talk about intestine particular because I mentioned they do have the pacemakers, they produce the pacemaker activity. Three types of potential: slow waves at rest, and spike with contractions, and these kind of spike with the contractions are seen. So here I could say parasympathetic stimulation, acetylcholine, stretch, cold, and all producing will influence the activity of the smooth muscle, and hormones like norepinephrine, epinephrine, and sympathetic stimulation also will influence the activity of the muscle. And in the mechanical contractions, as part of the contraction, some kind of the movements, mixing movement, small intestine, in the colon, this is called mixing, propulsive mixing movement for mixing the contents of the food with the gastric juice. And propulsive is pushing the food forward by forming a receptive relaxation. This is a receptive relaxation region, and food is pushed forward, seen in esophagus, stomach, and small intestine. These kind of movements, this kind of this kind of contractions are developed by production of three types of the action potential. We can ask why unstable resting membrane potential, why three type of the potential for making the movements, for producing the movements, and they are helpful in our body. With this positive note, I would like to close the lecture here. The smooth muscle, we have seen the structure, types of the smooth muscle fibers, and properties of the smooth muscle fiber, and with the innervation. So in this part, you can expect questions from this chapter, or one is contraction and relaxation of the smooth muscle, and the differences of the smooth muscle fibers, and the properties of the smooth muscle fibers are very important for examination point of view. So with another interesting topic, I will come in the next lecture. If you like my content, please like it, okay, subscribe and comment. Any doubts are there, so that immediately I respond. I will definitely answer for your questions. So thank you for patient listening of this lecture. Thanks a lot. Thank you so much.